Hello students, today we are going to continue with biology chapter number 4, that is molecular basis of inheritance. In this, till now we have studied the topic discovery of DNA. Then we have studied DNA as a genetic material with the help of different experiments such as Frederick-Griffith experiment, Avery-McLeod-McCarthy experiment, Hershey and Chase experiment. Then we have studied about packaging of DNA, why packaging of DNA is done, then packaging of DNA in prokaryotes and packaging of DNA in eukaryotes. Okay, then today we are going to study about the process of DNA replication. When we say study about DNA replication, we need to study first about the functions of DNA molecule. In this, the first function that we are going to study is DNA regulates and controls all the activities of the cell. That means all the metabolic activities of the cell are controlled and regulated with the help of DNA molecule. Then second function is it controls the synthesis of other molecules of the cell. That means the synthesis of the other molecules of the cell is controlled with the help of DNA molecule. Here the word synthesis means preparation. That means the preparation of other molecules of the cell is also controlled with the help of DNA molecule. The third function that we are going to study is during cell reproduction, DNA duplicate itself. This is something that is related to our topic that we are going to study in detail now. Okay, that means during the process of reproduction, the DNA duplicates itself. That means its replication process takes place during the process of reproduction. Then, next function is as a carrier of genetic information, DNA performs two important functions. One is heterocatalytic function and the other is autocatalytic function. When we say heterocatalytic function, it means that DNA directs the synthesis of chemical molecules other than itself. That means DNA is doing the synthesis of other molecules, leaving itself. It prepares these RNA molecules, it prepares these proteins. It means it regulates the synthesis of RNA, it regulates the synthesis of proteins. And when we say autocatalytic functions, that means it directs the synthesis of DNA itself. It directs the synthesis of DNA itself. Example for here is replication. Okay, when we say replication, that is our main topic for today that we are going to study in detail. First, the definition. We will see the definition of DNA replication. Here it means the process by which DNA duplicates itself is called replication. Okay, the process by which DNA duplicates itself is called as replication. In this process, the DNA molecule is duplicated. Okay, it undergoes the process of replication. After replication, it produces double copies which are exactly similar to itself. Now, before studying the process of replication, there are some key points that we need to remember here. That is, in replication, two identical copies are formed. That I have already discussed, that during the process of replication, two exactly similar copies are produced. In eukaryotic replication, the process takes place only one time in the cell cycle. That is, in eukaryotic organism, replication process takes place only once in the cell cycle. Then this whole process of replication occurs in S phase of the interphase. Okay, that we have studied what is S phase, that means synthesis phase. Okay, then it takes place in the nucleus. And the last point, that this process takes place through semi-conservative mode of replication. That was done with the help of Watson and Crick. We have already studied the Watson and Crick model of DNA. Now we are going to study the replication process, that is semi-conservative replication with the help of Watson and Crick. Okay, now we will start with the process. Mechanism of semi-conservative replication. When we say process, The other name for process is mechanism. This process or mechanism of semi-conservative replication we are going to study stepwise. Okay, in this the first step is activation of nucleotides. Okay, when we say activation of nucleotides, all the nucleotides, there are 4 types of nucleotides that we know, that adenine, guanine, cytosine, thymine. Okay, there are 4 types of nucleotides. This all nucleotides in their initial step are present in the monophosphate form, and they all are inactive. Okay, during the process of activation, they all are getting converted to their triphosphate form. That is, adenosine monophosphate gets converted to adenosine triphosphate, guanine monophosphate gets converted to guanine triphosphate, cytosine monophosphate gets converted to cytosine triphosphate, then thymine monophosphate gets converted to thymine triphosphate. And this process takes place in the presence of ATP and the enzyme phosphorylase, and hence this process is also known as phosphorylation. Okay, this process is known as phosphorylation. Now, moving to the next heading, next point, that is origin or initiation point. When we say origin or initiation point, this is the point where the process of replication begins, and this specific point is known as point O. Okay, the point where the process of replication begins is known as origin, and it is labeled as point O. And the unit of DNA in which replication occurs is called replicon. And with respect to cell, when we speak, the prokaryotic cells have only one replicon, whereas in eukaryotes there are several replicons. That means more than one replicon is present. At point O, enzyme endonuclease nicks one of the DNA strands. When I say nicks, it means it cuts one of the DNA strands. And then we will see what will happen after the cut. Okay. Third step is unwinding of DNA molecule. When the cut has been done with the help of endonuclease enzyme, after that cut, the DNA starts unwinding. Okay, and this unwinding is done with the help of the enzyme DNA helicase. When the DNA is unwinding, DNA molecule is unwinding, what will happen? It breaks the weak hydrogen bonds. Okay, it breaks the weak hydrogen bonds, and after the breaking of weak hydrogen bonds, what will happen? DNA will separate and unwind. Okay, now when it will separate and unwind, since it is a highly coiled structure, what will happen? Again it will get recoiled. There are possibilities of recoiling, but here there is something that is preventing from recoiling, and that is known as SSBP, that is single strand DNA binding protein. It prevents the DNA from recoiling. Okay, then moving on to the fourth point, that is replicating fork. Now see here in this diagram, replicating fork, I have labeled, this is labeled here as replicating fork. Okay, this is the point, origin point from where the replication process have started. So unwinding of DNA strands appear like a Y-shaped structure. You can see here it is inverted Y-shaped structure, and this is called as replicating fork. Okay, next point, synthesis of new strand. Now here when we say synthesis of new strand, see here in this diagram, synthesis of new strands. For this synthesis of new strand, the original strand, initial strand, the separated strand act as a template strand. You can see here in this diagram, here this is the original strand that has been resulted from the separation of DNA molecule, and these strands are called as template strand. Okay, for synthesis of new strand we require RNA primer. This RNA primers are attached at 3' end of the template strand. Then it is attached to the 3' end of the template strand. Okay, then RNA primer. As RNA primer attaches to the 3' end, what happens? It attracts the complementary nucleotides, and this all takes place under the influence of DNA polymerase. And one more point here, always new complementary strand is formed in 5' to 3' direction. You can see here, this is a new strand and it is formed in 5' to 3' direction. Again this side, new strand is formed in 5' to 3' direction. So this is all about synthesis of new strand. Then leading and lagging strand. Sixth point is leading and lagging strand. Now see here, this I will explain with the help of this diagram. The strand which has free 3' end. Okay, the strand which has free 3' end is known as leading template. This one is known as leading template, and its complementary strand that is newly synthesized. Okay, this is the complementary strand that is newly synthesized and it is known as leading strand, and this leading strand is continuous. Okay, and it is formed very fast. Then the strand which has free 5' end is known as lagging template, and the newly constructed strand that is newly synthesized is known as lagging strand, and but this lagging strand is discontinuous and it is formed in fragments. You can see here that this strand is formed in fragments, and these fragments are called as Okazaki fragments. Okay, these are called as Okazaki fragments, and it is named after the scientist Okazaki. These Okazaki fragments are later on joined with the help of the enzyme DNA ligase. Okay, they are later on joined with the help of the enzyme DNA ligase. Then the next step is formation of daughter DNA molecule. See here in this diagram that there are two daughter DNA molecules that are formed at the end of the process, in which you can see this pink line that is old strand and this blue one that is new strand. So here, see here, this new strand and the old strand as well as new strand, both are undergoing coiling with each other and results in the formation of daughter DNA molecule. Thus we can say that at the end of the process 50% is contributed by mother DNA and 50% is newly synthesized. Hence it is described as semi-conservative replication. Okay, now quickly we will recap all the points. First step was activation of nucleotide. Okay, and this what was done, the monophosphate form was got converted into the triphosphate form, and inactive nucleotides got converted into active nucleotides. Okay, second point is origin point or initiation point. This means the point from where the process of replication starts, that is the point O. And here there is a cut with the help of the enzyme endonuclease, that is called as NIC. Okay, then unwinding of DNA molecule takes place. Because of the cut, the DNA molecule starts unwinding, and this unwinding is done with the help of the enzyme helicase. Okay, and here one more enzyme is present. After unwinding, there are possibilities of coiling again, and that recoiling is prevented with the help of SSBP, that is single strand binding protein. Then replicating fork. At the point origin O, there is formation of inverted Y-shaped structure, fork-like structure. Therefore, that point is called as replicating fork. Then, after that separation, there is synthesis of new strand. This new strand synthesis takes place with the help of RNA primer. Okay, and the new strands are always synthesized in 5' to 3' direction. Then we have studied about leading strand and lagging strand. The old strand that are formed is known as template, and the new strand that are formed are known as strand, that is leading and lagging. Leading strand is continuous, lagging strand is discontinuous. lagging strand is formed in the form of Okazaki fragments, and those Okazaki fragments are later on joined with the help of enzyme DNA ligase. Then the last point is formation of daughter DNA molecule. Both the new strand and the old strand that are formed undergoes the process of coiling, and there is formation of daughter DNA molecule, in which one strand is old, one strand is new. Thus we say that 50% of the strand is formed from the mother molecule and 50% of the strand is newly constructed. Hence this process is known as semi-conservative replication. Thank you.